Thank you very much, Dr. Saidani, Dr. Kamani, and on behalf of Prince Vikram Data Mining Center, I welcome you all. Today we have a quite a long day and very interesting day with lots of talks. I would like to introduce our first speaker, Dr. Zaki, and please. Dr. Zaki is a professor of computer science at RPI. His research interests focused on developing novel data mining techniques, especially for applications in bioinformatics and social networks. He has published over 250 papers and chapters of data mining. He's a founding co-chair for BioKDD series of workshops. He's currently area editor for statistical analysis and data mining, and an associate editor for data mining and knowledge discovery. ECM transactions on knowledge discovery from data and social networks and mining. He has been the program co-chair for all the major data mining conferences, including ICBM, KDD, PAKDD, BIBM, and SGM. He's currently serving on the board of directors for ECM KDD. He received the National Science Foundation Career Award in 2001 and the Department of Energy Early Career Principal Investigator Award in 2002. He's also a senior member of IEEE and a distinguished ECM scientist. I'd like to welcome Mohammed Zaki, and we look forward to have a very exciting talk.
Good morning, everyone. It's a great honor and a pleasure for me to be here. Thank you for the invitation to be at the Prince Sultan University at this symposium on data mining and applications. So I am actually a professor at RPI, but currently I'm on leave at the Qatar Computing Research Institute in Doha. So the title of my talk is mining for patterns in today's complex data. And of course, there are many different applications of data mining in security, in health, in business analytics, business intelligence, and practically every field that we can think of. But some of the illustrations, some of the examples that I'm going to be showing will come from the field of bioinformatics. And that has also applications hopefully in the health community later on. But just to give a little bit more context and concrete examples, I'll be focusing on the application of these techniques within bioinformatics. So what has happened over the last 10, 20 years is the so-called inversion of science. So what used to happen in the past is that scientists in any given domain, they had a particular hypothesis in mind, and they used to collect data, and then they used to test, so with that hypothesis in mind, the data collection would happen, and then they would try to verify whether, what they were looking for was true or not, via hypothesis testing and so on. But then data collection became very cheap, right? Storage became extremely cheap, and people just started collecting data just for the sake of collecting data. And then suddenly it dawned that just this huge volume of data could have lots of value. So this actually started, I would say, maybe with the internet revolution. People just started collecting weblogs, and then people said, okay, can we use these weblogs to figure out, where people are visiting, which sites they're going, and Yahoo and Amazon, they started doing this product recommendations and so on. So I think the web revolution helped in this onset of big data that we are witnessing today. And the same thing has happened in the, in the bioinformatics community, and now it's happening in many other fields as well. And I think one of the most exciting things that's happening now is this notion of digital humanities. So traditionally, sciences have collected data, And if you talk to physicists, they've always been collecting big data. If you think about the super collider, from CERN, they collect petabytes of data, and they have been doing that in the past. But I think what's very new is, people from government, people from social sciences, now they have started collecting and analyzing these vast volumes of data. And this interplay between science, the humanities, social sciences, I think that will produce many interesting results in the coming years. So anyway, this has led to this, this realization that we need a new science of data. So across all the fields, are there commonalities that we can exploit? And so this is where of course, today's big buzzword is big data, and in some sense it's a buzzword, and in some sense it's a reality that we have to face, because the data is so large and it is extremely complex, and we need to make sense, look for correlations, look for patterns, build some interesting models to extract, useful insights. So for any given application domain, how can this interconnected nature of the different data sets be used to extract interesting, as they call it, nuggets of gold. So that's where the data mining comes in. You're sort of digging in this data for the nuggets of gold, or perhaps diamonds of insight. And the scale is extremely large, right? So you have highly uncertain, highly dynamic, and the data is obviously reaching petascales now, and it will even go on to exascale pretty soon. So there is a need to look at these kinds of data sets to extract knowledge. So of course, data mining has lots of different components to it. The part that I'm going to be focusing on is this notion of pattern extraction. So what do we mean by a pattern? A pattern is something that should happen more than once. It should actually happen often enough that we can make some, we can make some claims. A good example of a pattern would be, if you think of the online sites, let's say, to give you an example of Amazon, people go and buy books, and so a pattern might be that people who buy a book by one given author, they also buy a book by another author. Or if you're buying cameras, you're also buying, flash storage that goes along with that camera. And how often does this happen? So this notion of repetitiveness, that is something that happens frequently, is the sort of the fact that you'll be exploiting in extracting these kinds of patterns from data. And also, what you need is interestingness. So this sort of depends on the application. What are you trying to do with these patterns that you're mining? And you have to also talk about chance accuracy, because whenever the volume of data is extremely large, if you talk to any statistician, they will tell you that just by random chance, right, certain things are just bound to happen a certain number of times. So just because things are repetitive or frequent does not really mean that they are statistically significant. So you can always do this kind of testing to basically remove these things that just happen by chance in any large data set. And in general, what you want to do is that this pattern mining is what is called as an exploratory approach. So that is, you have a data set, you want to learn something, some correlations from that data, and then you can use these patterns. So once you know what's happening, some repetitive structures, you can use those to build other models. And these could be classification models where you're trying to say, this belongs to a certain group. What is the label of this item? You can cluster things, and you can build more complex models from these this kind of patterns that we expect. And then, like I said, pattern mining can be applied to many different domains, to e-commerce, to business. But I'll be illustrating some examples from bioinformatics because it's a very rich source of data. Lots of complex data sets have been collected, and so I'll be talking some patterns that we can see in these gene expression networks, in protein structures, and so we'll see examples of these applications in my talk. And one can obviously generalize these to many other application domains. So I hope you will see the connection that even though I'm talking about pattern mining in bioinformatics, In fact, the same concerns come up in social networks, the same concerns come up in, like I said, online recommendation systems. So the application domains are vast, but the underlying techniques are pretty much, they are in common. So let's start with some definitions. So what do I mean by a pattern? What kind of patterns can one extract? Now, of course, this is an open question. I mean, in some sense, it's like saying, I need a function to describe something, and what is the class of functions I'm going to consider? There are infinite number of possible ways of defining a function. You can have a linear function, a quadratic function, a cubic function, right? Polynomials, nonlinear functions, exponential, logarithmic, right? So the whole class is extremely vast. And likewise, you could say that when I want to look for patterns, the possible definitions of what is a pattern are infinite. And so we have to, of course, then say, okay, let's abstract and try to see what's common among these. And so here are some classes of patterns we're going to look at. So the simplest kind of a pattern is a set. So if you think of this A, B, C, and D, think of those as some labels. Maybe they are books by certain authors. So A is an item, B is another author, C and D are authors. And if I'm Amazon, then this is kind of like saying that this is an unordered collection, and I'm looking for this pattern. So people buy books by A, B, C, and D. Okay, so it's just a co-occurrence. So things that happen together. That's called a set. Of course, in the data mining community, they also call it an item set. So it's just co-occurrences. Of course, you can have sequences. You can add some sort of a dependence. You can say that actually, if I'm looking for a pattern, it's A followed by something else, followed by something else, followed by something else. And this can be true for, to give you an example from bioinformatics, in the genome, if you're looking for a pattern in the DNA, so the order matters, so which letter comes before another base, the DNA base, that governs how the functionality, for example. So in this case, we have a sequence. You can have more complex structures, how they are interconnected. And of course, the most general type of a pattern here is a graph. A graph is basically a set of nodes. They are connected by edges. So these could be different people, the different types of relationships with which they are connected. And each node and each label can have a label as well. Like what kind of a relationship is it? What kind of entity is it? So these are some classes of patterns. And then, I'm also going to talk about some other patterns shortly, which I call logical patterns, and we'll see some of the distinctions between them. So in my talk here, I'm going to be focusing on the graph class, because the graphs arise in social networks, in biological networks. Any time when you have an interconnected set of data with lots of links between them, and with lots of rich attributes, you want to discover patterns from that data set that can be modeled as a graph. And so one of the classes that we'll be looking at would be how to extract these interesting interconnected subgraphs from a large data set. So just to give you some examples of these, just to give a concrete example of sequences and trees and graphs, let me show you some illustrations from bioinformatics domain. So here's an example of a sequence pattern. So let me first focus on this one. So, when you have the human genome, what happens is that the human genome is like a database. It's a static entity, all your cells in your body have the same genome. However, the different contexts, the skin cells, the hair cells, they're doing different things because different parts of the genome are being expressed. So the genes, they get converted into proteins, and the proteins do all the work in the cell. And so here's an example of what we call a binding pattern, right? Something has to go. It's kind of like a machine that reads the database. So if I can use computer science terminology, so as a computer scientist, for me, biology is like an information system where the genome is the database. These proteins are actually molecular machines that go and bind. So here, in this double helical structure that you see is the DNA, and this, the spiral structure with some strands is actually a protein. It actually physically goes and binds to the DNA. So it's recognizing certain signals in that DNA. And what signal it is recognizing is something like this. So, you know, so this is a sequence of alphabets, you know, that are generalized. Normally there will be 4 bases, A, C, G, and T are the DNA letters, but we can generalize them and group them. And so this is saying that this particular, something called a transcription factor is a protein that goes and binds the DNA, and it has the sequential pattern. There is some signal followed by a gap between 5 and 180, you know, positions, and then there is another signal that it has to recognize. So that's an example of a sequence pattern that we can extract. And then the question is, you know, how can you use this? Of course, this could be used for maybe drug, you know, delivery to maybe remove an effect of maybe a bad gene, you know, that there is some mutation in the gene which is causing some disease. So if we know the signal, then we can disrupt that gene from being expressed. Right? So those are the kinds of ways that one can use once you can extract these patterns automatically. So let me move on. Another example of a pattern is from evolutionary tree. So, you know, when we look at, and this is the beauty of you know, God's creation, is that if you look at the genome of a human being, or a genome of a mouse, or a genome of a sperm whale, a lot of the genes are common. They're very similar. So in other words, you can infer some functionality. For example, you can do experiments on mice, and then you can see whether we can translate them to humans, right? So the reason we can do this is because these expressed part of the gene, they share certain patterns, they share certain, you know, sequences in common. And so people build these things called phylogenetic trees. That is an evolutionary tree that links together segments of the genes that are similar. And what they're showing is that, you know, there are many different ways of doing this, and of course no one knows what the right relationship is, but you can actually mine for the common, you know, this repetitive structure that you might see. So the idea would be, you know, you look at different organisms, you look at different genes, you then run this, you know, lots of algorithms on this data, and then from this data you can try to build something called a consensus tree. That is a frequently occurring subpart of the evolutionary, you know, network that is in common. And here's an example I'm showing, you know, a tree that you might discover. This is actually a plant family, and it turns out that if you run many of these algorithms, you can then figure out that this part seems to be in the same order many times. That means you have more confidence that this is the right, well, maybe not the right, but at least you have more confidence that this is the evolutionary relationship between these plant families. And moving on, you know, you can do this on the protein structures like I mentioned before. And this is where we go to the full, you know, general pattern of a graph. So here you have, you know, this structure that you see is actually a 3-dimensional protein molecule, and it has some interesting, you know, what we call secondary structures. So these, you know, the spiral structure is called an alpha helix, and these, you know, arrows that you see are called a beta strand. And you can represent these as actually graphs. You know, you can think of that as a 2-dimensional graph, even though the actual structure is 3D. But you can do a simpler representation like this, and then this can be converted into that format. And so you can see that this is actually nothing but, you know, in computational terms, it's a graph, and you can now look for common patterns among a class of proteins. And here I'm showing that, for example, if you look at these 3 protein molecules, they have obviously certain similarities in common. That even just looking at them, you can see that, for example, this structure here appeared many times in that one. And so you can again look for these common, what in bioinformatics we call them motifs. And this could be a good signal, again, you know, if you're designing drugs for you know, hard problems where protein structure plays a role, you can use this kind of, you know, motif extraction techniques to target drugs appropriately to the different application. And then, of course, there are many other, you know, there are interaction networks between proteins, and you want to extract, again, graphs from those. And I'll just, you know, go very quickly through these. You can do this on, you know, tissue cultures, you know, where in this example I'm showing, you know, this is actually from, so data from cancer. So this is actually healthy tissue, and this is a disease, like a cancerous tissue, and there's something else called an inflammation. So this is actually not cancer, but it is some sort of intermediate, it's not really, it's basically a benign state. And so you want to sort of figure that out, but you want to basically distinguish between healthy and diseased tissues. Now, one way to do this is you can actually model these as graphs, and then you can, again, do graph mining on that. So again, there are a lot of, the applications of graph mining are pretty much from the biomedical to the bioinformatics side. One can use these structures to mine patterns. And I'll just skip through this as other examples of graphs that appear at metabolic pathways, different biological processes, and so on. So that was one class of patterns. So I mentioned, sequences, sets, sequences, trees, and graphs. Now let me just switch gears and talk a little bit about what I call logical patterns. So as I said, the class of patterns can be wide. You have to focus on certain of these classes. So here are what I call logical or Boolean patterns. So remember the set I showed you before. A set is simply a conjunction. It's like something happens and something else happens and something else happens and something else happens. So in this case, you can think of this as what we call an AND clause. That is, the actual connectivity between those elements of a set is the relationship AND. A and B and C and D. So this hat that you see here, or the wedge, is what I'm using for AND. So that's called a conjunction. On the other hand, you might say, okay, why not look for disjunction? Can I look for A or B or C or D? That means any one of those things that happens is interesting. So that is what is known as an OR clause or disjunction. And this would be, the inverted V structure I use here as an OR. So this is saying that I'm looking for these patterns where any one of these things happens is interesting. And then you can also connect them in many other complex ways. And there are these so-called normal forms that people have defined. So one of them is the conjunctive normal form. Let me just focus on the DNF, the disjunctive normal form, which is a pattern like this. So A and B happen, or C and D happen. That is, this is the kind of pattern you're looking for. And this again has many applications, as you'll see. You can use these. So let me just skip through this. I don't want to. This was more of a extended. So let me go to the application here. So where would you use such a sort of this Boolean or logical pattern? Why would it be interesting? So one example of this is the so-called gene expression behavior. So I told you about genes, right? So genes are being expressed, meaning being converted into proteins, and these proteins are doing all the work. Now people want to know, let's say between cancer and healthy states, it is known that different genes are expressed when a tissue is diseased, and different proteins are expressed when the tissue is healthy. Now, how do you measure this? Right? At the molecular level, that is at the level of proteins and genes, they have this technology called microarrays, the DNA microarray technology. It looks like a chip. So this is actually one of the images that one gets. So basically, you take a tissue sample, and you basically put this through a small chip, actually, and you basically measure each dot here. Okay, I mean, it's a little bit small, perhaps it's hard to see in the back, but each dot in this array is telling us which gene is present in which quantity. Okay, so the brighter red, or the more green it is, the more highly expressed it is. And if it's black, that means it's neutral. If it's red, it means it's underexpressed. So this kind of an image can be converted into a matrix. So what you have is, you have a bunch of genes, a gene 1, gene 2, in this case, it's a small example, there are only 5 genes. And what you do is you measure how much of that gene is present under different conditions. So under cancer conditions, under healthy conditions, under other types of conditions that you might have from different patients, for example. So these I call samples, sample 1, sample 2, sample 3, and so on. So this matrix is what you need to analyze. And what people normally do is they look at the expression level, that is the amount of that gene that is present, and they make it into a binary value. They say, if you very highly express, that's 1, otherwise it's zero. So think of this value here, the blue boxes, think of those as basically presence. And if it's white, means that gene is not expressed. So the interpretation would be gene 1 is not present in any of the 5 samples. Gene 2 is not present in sample 1, but it's present in sample 2, present in sample 3, present in sample 5, like that. And now you have actually a binary data set, a binary matrix, and here you can actually come up with patterns like, certain genes are present or not. So I can say gene 2 and gene 5, gene 4 and gene 5 are present in sample 2, sample 3, sample 5. And that would be an example of a Boolean or a logical pattern that you can mine. And so this is actually what I had here, was to show some example of patterns that one may extract, and so on and so forth. So again, to show you that there is a rich class of applications for these kinds of patterns, from graphs to Boolean patterns. So I'll just skip over this in the interest of time, and go on to some maybe techniques. So the core part of my talk is that there are lots of these pattern classes, and you can see the data is pretty complex, right? Sometimes it is the whole genome, sometimes it is the protein structure, sometimes it is some sort of an interaction network. And as I said, the applications can come from a totally different area. Let's talk about social networks, right? So let's talk about Facebook, let's talk about LinkedIn, right? Let's talk about online sites like Amazon. There also you have the same phenomenon. You have friendship networks in Facebook, for example, or you have Twitter networks, people tweeting things, retweeting things. So this would be like a news or a information dissemination network. So again, the graphs come up in many different domains, not just in bio, but in all these other applications as well. So you could say, okay, how should I go about mining these patterns, right? So what are the techniques that people use? So let me give you a brief illustration of how people do it. And given the scale, then I'll justify why we need sampling, because it turns out that the data is so large that you cannot hope to extract everything in a complete way, because then the complexity of the problem is known to be NP hard. It's a very hard problem, and the exponential number of patterns you are producing. So obviously, you are not going to be able to mine everything. So then we sample, and the question then is, how do you sample well? So let me give you an illustration, just to give you again a concrete example. Let's just take a very simple example. Here I have these 3 graphs. Okay, graph 1, as you can see, the labeled interconnected structure. You have different labels that you repeat. Here's another graph, here's another graph. And so I have a very simple question. The question is, I want to find patterns that appear in 2 out of the 3 graphs. So this is the user condition. So this is my criteria. I'm saying that if something happens 2 times out of the 3, then it's interesting to me for some reason. Okay, can you extract the pattern that happened 2 times out of 3? So an example of a pattern would be this. So this is actually one of the largest patterns you can extract. And this happens 2 times, which is shown in the red, sort of how does this pattern appear in these input graphs, the 3 graphs. So you can see that this structure AA connected to the second A should be connected to the C and the D. That happens here, and it also happens here. And so this would be an example of a pattern that appears 2 times out of 3. Another pattern that appears 2 times is this one, and this happens in graph number 1 and graph number 3. Right, so this is the kind of thing that we want to do. We want to be able to extract these subgraphs from a collection of graphs. And so this is something called the support. The support is how many occurrences are we looking for. In this case, we're saying we're looking for 2 occurrences, so I call that the minimum support threshold. So someone gives us that threshold value, and we mine it. And so these are, in fact, the 2 largest patterns we can extract. So these are also called the maximal patterns. So the definition of maximal means there is no larger pattern that is frequent. So if I add another label, add another vertex or another edge, the pattern will no longer appear 2 times. Okay? The frequency will go down. So you want to be able to extract these patterns. And here's an example of like in a chemical compound. So think of these as maybe compounds that target a particular protein. Maybe it is for cancer fighting drugs. And you also want to know what's common among these drugs, right? So which are the substructures that happen in common. So in this, you have different drugs, and then what is common to them, of course, you can see that this ring is in common, like it appears here, it appears here, and it appears here as well. And then there's another substructure that, so people actually use these techniques quite often in drug design, in drug discovery, to extract these frequent patterns from it. And another example, for another flavor of this problem is when you have instead of, so if you go back a second here, I had 3 different graphs. So this would, for example, the 3 different chemical compounds. But often what happens is that you have just one single graph. So this would be an example of maybe the Facebook network. Or in the case of bioinformatics, it could be a protein interaction network or a gene network. So there's only one single network, very large. In the case of Facebook, it's like 1 billion users, with billions of edges. And now you still want to find frequent patterns. And so in this case, you could again look for this pattern here. But now what happens is that there's only a single graph, and you have to look for all possible ways in which this subgraph can appear. And here are some of the ways. So there's 1, that's 2, 3, 4, 5, and 6. So it turns out that this is the so-called total number of occurrences of this pattern is 6. And we're looking for these kinds of things. So anyway, I'm going to skip through this. So what people normally do is when you want to extract these frequent patterns, so let me go back to this example of 3 input data sets, and you're looking for frequent things. What people do is they do what is known as a complete search. So complete search says, I'm going to start with single edges, and I'm going to grow the pattern 1 edge at a time. So I'm then going to look for patterns with 2 edges, with 3 edges, and so on. And each time, I'm going to compute the frequency. So in this case, you have these single edges that might be frequent. You pick one of them, and you expand them. Here are all the possible ways in which you can extend this by 1 more edge. So how can I take A and C and add 1 more edge? I can add an edge to the C. So in this case, you can see from the C, I have another edge, labeled A, B, C, and D. Or you can take that first A and extend from that. So here I see I can extend to the A, to the B, to the C, and the D. Because there are 4 labels. I'm assuming there are only 4 labels. And here are all the ways that I can extend it. And then you count again. You say, okay, how many of these extensions are frequent? Okay, so you count those that are not frequent, you throw away. So in this case, you throw that out because it's not frequent, not frequent, not frequent, not frequent. Anything that survives is frequent, and you go on and extend it. So that's normally how it works. So people can design very fast ways of pruning things. So the idea is that the search space is exponential, and you want to be able to prune it very quickly. And this is what is known as complete search. You're searching everything that meets your criteria. So the criteria here is just a frequency criteria. Things must happen 2 times for it to be interesting. The problem here is that this is a hard problem. It's a known hard problem. And there are these 2 problems we have to tackle. One is known as graph isomorphism, that when can I know 2 graphs are duplicates? Because you don't want to mine the same pattern repeatedly. Once you found it, you should be able to say, I found this pattern, I don't need to discover it again. But it turns out that in graphs, this is a hard problem. Actually, no one knows whether this problem can be solved in polynomial time. This is a currently open question, whether you can detect whether 2 graphs are the same in polynomial time. And of course, the other problem is much harder, that is when you try to count. So given graph, how many times it happens in my data set, that is actually known as subgraph isomorphism, and that is a known NP-hard problem. So the complexity is extremely high, and so you have to run this if you do this. So the idea, so the problem is that you have to do a lot of these checks. Every time you have a large data set, you're doing millions of checks for subgraph isomorphism, and that is essentially what kills the problem in real application. So let me give you an example. So what we did is we took a real-life data set. So let me go back to this one. Here you have that, so let's say you're trying to find out, remember this is the healthy tissue, this is the cancer tissue. Let's say I want to build a classifier. A classifier is a model that says, can I distinguish between healthy and disease states, right? And actually in this case, there is a confounding a tissue sample as well, which is a damaged state. So you want to distinguish between these three classes. Now, the point is, what we want to do is we want to look for the so-called discriminatory subgraphs. That is, since I'm doing a graph modeling of these tissues, can I come up with a graph that appears only in healthy and not in cancer, or it appears only in cancer state but not in healthy or damaged? So you're looking for these patterns that can distinguish one state from another state, right? So you want to do graph mining to extract this. It turns out that we took these like just 30 of these tissue samples. Each of these had about 2,000 entities or nodes and about 36,000 edges, and you try to mine this. And then we made some sort of minimum support of 40% of my tissues should have this pattern. And it turns out that we were not actually able to mine using some of the state-of-the-art techniques. Of course, this is on a single machine and so on and so forth. You can obviously make it parallel and maybe try to scale it up. But the point is that state-of-the-art methods basically failed to produce any output because these are very large graphs, as you can see, and you're looking for frequent things. So, and the pattern itself is very large. And then we did the same thing for another application where you're looking for protein networks from different sources. And again, in this, what happened is that essentially it gave us some output, but it gave us too much output. So, so starting from 3 graphs, in this case there were about 2,000 nodes and about 80,000 edges in the whole set of graphs. And here what happened is that we actually got too much of an output. So we ran this for 1 day, and we got like 8 million subgraphs were put out. So here's the opposite problem. You have like information overload. You start from a small enough data set, but then you get so much, so many patterns. What do you do with it? So then we said, okay, so this is not really working out. So if you try to mine everything completely, right, things are extremely expensive. So can we speed things up? So we said, okay, let's sample. And so let me just give you an idea of sampling. So it turns out that these problems can be solved by sampling much faster. And so here's the basic idea. So this is actually the same data set that I showed you before. And here's a very simple idea, and one can improve upon this as I'll show later. So the idea is that I pick any edge and test whether it is frequent. So in this case, I'll start with AA, and I count. Okay, AA happens over here, AA happens over here, and my minimum frequency is 2. So I'm looking for an occurrence of 2 out of 3 times. And it turns out that I got lucky this time, and this actually is frequent. So, sorry, this should be frequent. It appears 2 times. Okay, so what you do then is you just do a random extension. So pick any one of these vertices, maybe this one or this one, and extend by 1 more edge. So we tried this one. So we said, okay, let's extend this node by the label B. And then you see, okay, does it appear here? Yes. Does it appear here? No, because you don't see any AA, there's no B connected to any one of those. And neither does it happen over here. So in this case, we have to reject it because this move, so to speak, is not valid, right? It's not frequent. So we say, okay, this is not a fruitful extension. Let's simply discard that and try something else. Okay, so then you move on. Do a random extension from the second edge, from the second node. You do the extension to the C. Oh, and this time it turns out to be frequent. So you see what we're doing is basically extending randomly. We are counting, and then we're testing whether it's frequent or not. If it's frequent, you extend more. So you keep on climbing up in the search space very quickly. So this is a real advantage. You keep on doing this, try something not frequent, then throw it away. Try something else, not frequent. Try something else. Oh, it's frequent. That's great. You keep it and keep on doing this till you cannot add anything else. And once you cannot add anything else, by definition, that is a maximal pattern. Okay, because maximality means that I cannot add any other label, edge, and yet it will be frequent. So in this case, we actually try all possible extensions, and we find out, okay, I cannot add anything else at all. That means I'm finished. This is one of the patterns that I find, and one of the things I showed before. Of course, the structure here is slightly different, but this is one of the maximal patterns that we found. So you can see the power of this method, at least intuitively, that you're climbing up very rapidly in the search space just by a random extension. Whereas if you do complete search, you do level 1, level 2, level 3. By the time I get to level 100, that would be more than the age of the universe, because you'd have to enumerate 2 to the power 100 subgraphs before you can get to a level 100 pattern. But in this, you can do this in 100 steps. So that essentially is the power of this kind of sampling. So let me just skip through these, because these are essentially detailed and I'm running out of time. But this illustrates the power of the method. So one of those graphs that I showed you, on which I said that we had like 7 gigabyte of output with 8 million subgraphs in a single day, and it was not even finished. It was still churning out. It turns out that this dataset you can actually mine for all the maximal patterns via sampling in just 450 seconds. So instead of running this with so many different things, and this is kind of like the proof of the method. So in this chart, what you see is the number of random extensions that we did. So from 0, about 400 walks, 800 walks, 1200 walks, to about 2000 walks. And on the y-axis, I'm showing how many unique patterns were found. So you see, this curve is showing you how many unique patterns were found. So initially, as you add more random walks, you discover new and new patterns. And so you keep on finding new things. And then at about 400, you're almost at the knee of the curve. By about 1200, you basically found almost everything that is to be found. Because after that, right, more walks are not adding new patterns to your search space. That means you're not finding anything new. Of course, you might say, how do I know that I've finished, right? So that's always the question. In this particular data set, I knew exactly how many patterns there were, and I could claim that there were only 90 maximal subgraphs, and we found all of them. But in general, I would, be concerned that actually I do not know when I'm finished. So what you can do is you can monitor the rate of new patterns that are being produced, and you can stop at some point when you are sort of, when it sort of tails off. That means more walks are not adding new patterns. In fact, that is one of the criticisms of this method is that what I've just shown you is what is called as a non-uniform walk. That means there is no statistical guarantee of whether I have converged, whether the sample I've given you is representative or not. And there are methods to fix this, so that's good news. So this method is very fast, but it doesn't give you any statistical guarantee of sampling. It turns out that one can do more powerful methods called Markov chain Monte Carlo methods. So these are well-known methods in statistical physics and other domains. And you can use these sampling techniques to actually give a guarantee for that actually sample in my space. So in the interest of time, I'm just going to just say that these methods exist, and they give guarantees. And what is interesting about these methods is that they allow you to choose different kinds of sampling, not just uniform sampling. So the first thing one should do is, you look at the whole possible space of patterns that one could mine, and the simplest thing I could do is I could say, give me a uniform sample from that space. Right? So this is a very large space, it's exponentially large, and I want to sample patterns from this space. So that would be an example of uniform sampling. Well, that's good, and the methods can do that. But sometimes you want to find those patterns that are more frequent, you should sample them more. So you can do what is known as support bias sampling. You can also do discriminatory subgraph sampling. So this would be very important for that cancer versus healthy problem that I mentioned before. Because here we are looking for a particular type of a graph pattern. A pattern that appears frequently in the cancer state, but not in any of the other states, or it happens frequently in the healthy state, but not in any of the other states. Right? So you try to look for discriminatory subgraphs, and this can also be embedded in the search itself. So when you're searching, you're only looking for those kind of patterns, and that becomes what is known as a quality measure, like a probability distribution of the pattern, and you sample according to your underlying distribution that you're looking for. In this case, you would sample everything uniformly. Everything is just 1/n. It's a uniform distribution. In this case, it would be a support bias distribution. So if one pattern happens 10 times, another pattern happens only 5 times, right? The pattern that happens 10 times should be sampled twice as more as the one that happens 5 times. So that becomes the probability distribution over the pattern space. And here also you have to define an appropriate target, distribution that you desire. and you can do other things as well. So this is the generality of the method, is that it can be applied to many different sampling tasks, many different interesting tasks. So yeah, I'm gonna skip through these. These are just more details. So this is the bottom line in that is that once you do this kind of NCMC and other, one can use other sampling methods like importance sampling, gift sampling, and so on. That's good because they give you a statistical guarantee. But unfortunately, efficiency then goes down, because what happens is these methods randomly walk in the space, and then it takes much more time. And so there is still a lot of work to be done in improving this whole approach. So on the one hand, there's no guarantee, but it's fast. In this case, there is a guarantee, but they're slow. So how do we, merge those two? So yeah, I'm going to skip through these because I don't have time. Basically, I want to jump to the concluding part of my talk, which is basically this is where we are. We have today dealt with basically two main problems that I see in terms of data. The data is very complex and it is very large. So the scalability and the complexity are the two twin sort of, if I think of this as a monster that has to be tamed, it has these two heads. So complexity basically means that you have lots of interesting types of things happening, lots of uncertainty in the data that you have, lots of dynamic stuff. And then the scalability says, how do I deal with petascale, exascale data, right? So that's the real challenge. How do we summarize quickly? And we need these kinds of methods. So sampling is a good way, but there are many other ways of, people talk about sublinear algorithms or one-pass algorithms, streaming methods. But these are issues that are going to be very important in the future. How do we do uncertain data? Every data set that we have is uncertain. There is no guarantee that what we see is what is actually in the data. So one has to design methods for approximation. And this part is currently under study. Okay, so for those PhD students who are interested or master's students, it's a very important topic to work on is uncertain data. How do we handle that? And then just in the case of biology, there are lots of interesting sources, and the same thing is true for social networks and other interesting data sets. So essentially, that's why I'd like to conclude here by saying that there are lots of opportunities, and we're just beginning to sort of scratch the surface of this field. Thank you.
We can take questions from the audience now.
Thank you. Thank you. Excellent. I have two questions. The first one is the first approach that you present when you do some kind
Extension.
And extension. It looks like it may be started in local minimum because you choose randomly one node, and this is why, of course, the local minimum we can do very quickly, and we will be steady in kind of plateau. This is maybe the issue when we have it.
That's an excellent question, actually. It turns out, as you correctly point out, that whenever you're searching a very complex search space, you are not sure whether you are in one part of the search space, and they might be this whole other part, but there's only a very narrow link between them. So this is actually something called the mixing rate of the Markov chain. Because if you think of this, in technical terms, it's kind of jumping from one state to the next. So it's and it depends only on the previous state. So it's a Markov process, and this is a real issue. So unfortunately, these are tough problems, and it is possible that you might get stuck because you might be searching this huge thing over here, and you cannot get to this part because there's a very narrow link here. And there are techniques to jump across, and I would say in this case, what people normally do is you can use genetic algorithms. So there are many other search techniques one can throw on top of this to enable this kind of sampling from start from different random states and explore. And then at some point you have to sort of say that but that's
Second part maybe let's say when you apply MCNC, which provide very nice result. How you think that you can do parallel with, let's say, the social network? So, or for streaming data. for security issues, most probably we do surveillance of data online, real time. How we can apply this kind of approach, or at least a variant of it, to do this on stream?
Yes. So there are actually, okay, so in terms of parallelism, the good news is that these are extremely hard problems. So when I'm computing the frequency of a graph in a big dataset, that step itself can be made parallel. But on the other hand, you can also run many different walks at the same time. And those are completely, what is known as embarrassingly parallel. So I can run thousands of these walks at the same time. So you can actually leverage parallel computing very nicely in these problems.
Because in streaming, you don't have, in this approach, we have the data set.
Oh, the streaming is much more difficult. Yeah,
we don't have it, so I don't know how we will be tuned to fit this streaming data.
To be honest, one has to also scale down our expectations. So my experience says that when you have a large volume of data, you should first try to live with simpler patterns. So don't try to mine graphs before you mine sets, because you might get enough valuable information just from co-occurrences. And there are methods that are very fast for sets. You can summarize, you can look for hot items and so on. And you should only go to graph if there's a real need for it. And I think in the streaming case, if I have I think some of these, I would be skeptical, but of course, one would develop methods for it, because there might be applications where you need it. But they are very challenging. I agree. And it's open, I think it's an open question right now.
Thank you, I appreciate it.
And I also thank you, Professor Zaki, for this insightful presentation. My question is conceptual rather than in the concept of data-driven science as opposed to theory-guided science. How can you see the disadvantage of guiding the science through the data rather than guiding science through the theories? Can you comment on this?
So I see this actually as the flip side of a coin, in the sense that normally what you want to do is you want to merge these two fields together. So what has happened in the past is that scientists, so let's say if I'm a climatologist, I want to see weather patterns and over a long period of time. I can start with hypothesis and I can collect some data. Normally what I'll do is I'll run some simulations of my model. Now it turns out that what we want to do is we want to look at the data to guide and build us better models. So what you want to do is you actually want to close the loop. That is, all this data that's being collected, how can it help us enable, rather how can it enable better informed models? So I think this is currently there's a gap between the world of simulation and the world of data. So we do data mining, we extract all these patterns, and no one does anything with it. On the other side, you have the classical, like where they simulate a lot of these materials, climate science, many other domains, and they are also producing a lot of data. The simulation data is extremely large, and one can actually mine from that and then rebuild the model or closing the loop. And so I think that's one of the opportunities, is that currently it's not happening, but I think people are beginning to realize that there is a sort of there has to be this interplay between the two sides, that data can inform and help us build better models, and better models will produce a lot of the simulation data that we can still mine again. So I basically see in that context that I don't think they are separate things. Data mining is there to help us come up with hypotheses, which should then be verified. So in some sense, what you're doing is you're using data mining as a tool to generate these hypotheses automatically. And then you would test each of these hypotheses with a more domain theory, with the actual domain scientists. And so we need to work together. That's my take on that.
Take a question from our colleagues on the lady side. Any questions there? Do we have a question on the lady side? So can you go ahead, one more question?
First, about 10 not happening.
We can't hear you very well. Can you please increase the volume?
I would like first to thank Mr. Zaki for the great talk. Here we talked about the graph mining pattern, pattern mining, and he really gave an example for bioinformatics. And here we give a chance for applying the graph mining for certain methods. Can you please give us some examples of how it can apply in social methods? And what is the current problems for the social networks, and what tools can we use it for the graph mining?
Okay, thank you. So for social networks, I can give you an example. So one very good use of graph pattern mining in the context of social networks would be, so if you're aware of the current trend, right? So Facebook has started this new thing called social recommendation. So the idea is that, let's say, I have my colleagues, I have my family, I may have other friends. So I have professional friends, my casual friends, let's say recreational, and then I have my family circle. And now, what can I do with this information? Of course, Facebook, for example, as a company, has information about this from many different people, right? So what they do is they say, okay, let's look at, so if you think of the graph, each node is a person, and the connections are, how am I connected to this person? Is it because I play squash with them, or is it because we are in the same university? So these kinds of links can then be exploited, and then I look at their interests. So maybe I have a list of interests. Okay, so I'm interested in, these kind of activities. So these become the labels. And the idea then is, so I have my own, small network. Everyone else has their own small network. Can I look for common patterns among them, right? So, and then how can I use it? I can use, so once I can extract these subgraphs, which are the sort of, let's call them interest-specific subgraphs, then they allow us to make recommendations. So actually, from a Facebook point of view, they want to sell us, products, which may not be the most interesting, societal benefit, but on the other hand, it has tremendous value, because you say you want to buy this, your friends might be likely to buy this product, or or just that you should connect with this other person. It looks like your friendship network and your interests are similar to this other person. So so these companies are using these kinds of networks to basically mine these patterns. The same thing can be done on a Twitter network. So in this case, what you're doing is you get some a tweet, you retweet it, and you have this followee kind of the retweet network, so to speak, and people want to predict which item will become more popular. Is it does it depend on what labels you're using? What are the hashtags that that people are using? So you can again do mining to figure out what kinds of hashtags, for example, trend more. People they get very, very popular. So they have lots of applications in these kind of networks.
Thank you, Dr. Zaki. I'm sure there's going to be a lot more questions. I'd like to invite you to post a session we're going to have during the second refreshment break, so you can connect with our invited speakers. I'd like to invite Dr. Yanai, who is now on the stage. He would like to present a token for initiation to Dr. Zaki.